Good morning, everybody, and thank you for joining us today for this webinar on spectral imaging in plant sciences. I'm going to concentrate mostly on multispectral imaging, and you'll see a lot of examples later using the VideometerLab 4 instrument you can see here. But I will start off, first of all, by talking a bit about the background to spectral imaging. And where it comes from. I'll tell you a little bit about the kind of hyperspectral imaging hardware that Analytik can supply from Headwall Photonics. I'll show you a few examples of precision agricultural applications for hyperspectral imaging. Then I'll move on to a bit about multispectral imaging versus hyperspectral. Talk a little bit more about the VideometerLab that you can see here and some of the applications we've developed with that, specifically in plant sciences, and I'll show you a few kind of cool things. So if we start the presentation. So they say a picture is worth 1,000 words. It's a well-known phrase, and by analogy a spectral image is worth 1,000 pictures. So we can see here an example of hyperspectral data cube, or hypercube, they're sometimes known as. The images here, you can actually see a body of water, perhaps a town of some sort just here, and some coastline. And behind each pixel in this image is an entire spectrum collected for each pixel. And there's a huge amount of information in there that we can use to tell a lot about the scene. So what is spectral imaging in the first place? Well, it's a hybrid of spectroscopy and imaging. So we've had these two techniques, spectroscopy and imaging, have been around for well over 100 years, each of them. On the left-hand side, you can see a spectrograph of light collected from an exoplanet, actually, a planet in another solar system. And we can tell a lot about the atmosphere on this planet just by looking at the light collected from when it passes in front of its home star. So all these gaps in the spectrum tell us a lot about the chemical composition of its atmosphere. And of course here we have an oldie-timey black and white image. Imaging has been around for a long time, and ever since it came about, we've been using images to measure stuff, photogrammetry. Imaging has developed from black and white into red, green, blue color images designed for our own eyes, and then the next evolution up from that, of course, is spectral imaging. So spectral imaging is much better, if you like, than RGB color, which in turn is much better than monochrome. We're getting more and more information with each evolution in the chain. And so what we get with a spectral image is the best of both worlds, the best of spectroscopy and imaging. So with spectroscopy, we get a lot of information about the chemical identity, but we lose any information about spatial location. We just measure one spot. Whereas with RGB imaging or monochrome imaging, we have our spatial location information, but we don't really have any information about the chemical identity. A spectral image, on the other hand, at the far right, we have both the chemical identity and the spatial location in the image. So this technique is, has a few names depending on your background. It's known as hyperspectral imaging or imaging spectroscopy, sometimes chemical imaging. It depends what field you're talking about. They can all be kind of grouped together, and they all use pretty much the same kind of hardware. Here's an example of a hyperspectral imaging spectrometer from Headwall Photonics, and I'll go over a bit more later about how that works. Then multispectral imaging is really the same idea as hyperspectral imaging, the same technique, except it's much more cost effective, it's a lot easier to do, a lot easier to analyze, and I'll tell you in a bit about why that is. So just to take a quick step backwards and go back to basics, apologies for those of you who are well familiar with this, but just to cover all bases. Here we have an instrument on the left here. It's called the Handheld 2. It's from a supplier of ours called ASD, the ASD Handheld 2. And you can see what we have here is a probe that has an integrated light source. It's just a quartz tungsten halogen bulb and a fiber optic cable to collect light that reflects off the leaf. So it illuminates the leaf, in a certain area with the light source. Then we choose an area somewhere in the middle to measure, and we collect that light that comes back into the fiber optic probe, comes back up and into the unit where it's analyzed. This is a visual range light, and we get an awful lot of information from that. This is a widely used technique in all sorts of areas. You get a spectral signature for that leaf. You can see here the green line represents a green leaf, and the red line is a red leaf, and we can tell whether it's red or green from the spectral signature. The idea, the drawback, I guess, for spectroscopy is that you are only collecting information from one average area. So that spot could be very small, just a millimeter across, could be very large, it could be a couple of centimeters across, but it's always going to be the average of everything in that area. The idea with imaging spectroscopy or hyperspectral imaging is that we're taking many data points over the area. So here we can imagine we've used the ASD handheld to take an 8x8 picture, 8 pixels wide and 8 pixels deep image with a spectra behind each pixel. The only drawback to doing this is it would be really tedious and slow to do. And so luckily we can do it with specialist hardware. Now, the idea of hyperspectral imaging has actually been around for decades, mostly employed on satellites and airborne vehicles. So what goes on here is we have the sun as a light source, and then the satellite or airborne vehicle will move over the scene and collect data as it goes. So this image on the right here, this is a NASA JPL Avaris craft. Avaris stands for Airborne Visible and Infrared Imaging Spectrometer, and the kind of data it collects will look like this. So what we have, these are 2 data cubes collected with Avaris. You can see on the left we have a scene of perhaps some buildings down at the bottom left, a body of water, a road, and behind each of these pixels will be a very high resolution spectrum, a spectral signature for that area of the ground that the pixel is covering. The width of the image is going to be determined by how wide your sensor is, and the length can be as long as you like, because as you fly along, as you fly over the scene, it just collects more and more data, and it adds line by line to the data cube, a bit like an inkjet printer, and it builds up the image that way. Just a quick word about the actual data you get from a hyperspectral camera. You have to analyze it using multivariate statistics in specialist software packages. So things like Envy, which has been around for a very long time. There's others like Gerbil Optics. You can use MATLAB, and it involves very big data files. So you're talking gigabytes and gigabytes of data per data cube, requiring very powerful computers, very complex analysis. Just to reiterate, there's kind of 2 ways to look at a data cube. Here we have, again, an image of some fields, a bit of woodland on the left. What you can look at is perhaps 1 pixel and all the wavelengths in that pixel. We have a nice smooth spectrum, spectral signature in 1 pixel. Or we could look at all the pixels, the whole area, but at 1 single wavelength. So this image could be specifically at green 550 nanometers light. So there's 2 ways of looking at a data cube. And what are they being used for nowadays? Well, precision agriculture is the dominating application, really. It's used for looking at crop stresses, so for nutrients, water, disease and pests, soil characterization, vegetative cover and yield estimation. But it's a very widely applicable technique. It's very versatile. It's used for environmental monitoring, so that's largely what these satellites are doing. When they have their hyperspectral or multispectral cameras, they'll be flying over disaster areas, such as in Canada at the moment. The Avaris aircraft has been used to look at the wildfires going on. It can also be used for food quality and authenticity, contamination. So the Food Standards Agency is at the moment very interested in the potential for hyperspectral and multispectral imaging for looking at food authenticity in particular. It has applications in pharmaceuticals as well, so looking at coatings on tablets or mixtures of powders, and you can actually mount these on a production line. They're very well suited to looking at moving webs. So for example, the satellite or plane has to fly over the scene to collect the image, but you could also have a product moving under the camera instead. And of course, finally, military and defense applications. In fact, military and defense was in the past, until the past few years, it was the main market for these type of cameras. They obviously had the money to spend and the top secret applications, but precision agriculture has taken over by a long margin now over military and defense. Precision agriculture is really the main driver for this type of imaging application. So here we can see an example of a head wall photonics hyperspectral imaging spectrograph. So what you can see is it's very compact, it's quite small actually, the way that the head wall design works. Down in the bottom right we have a schematic of how they work. What happens is you have a slit here, a very fine slit, so that's the width of the sensor, and we're going to be collecting one line of the image, one line at a time, and that may be, say, 1,000 pixels wide. You'll get a 1,000 pixel wide image. Here we have the light path for 1 pixel coming into the system. It hits a focusing mirror, goes to a grating, so this is where Headwall's expertise is, and is in very high quality, very carefully designed diffraction gratings, very much like a CD. When you see the reflection of a CD, it's diffracting the light, and it separates the light into its red and blue and green components, which then are then focused onto an array, a sensor array, a fairly normal sensor array, really, much a bit like you'd find in your phone. So these are the types of sensors that have been for a long time put up in satellites and on airborne craft, and over the years, what with drones coming around, Headwall have put a lot of work into developing incredible piece of engineering, really. This is called the Nano Hyperspec here. You can see on the left here a golf ball just next to it, so you can see just how tiny it is. So all those focusing optics are all inside here. You get a very high signal to noise ratio, very high quality, but it's tiny. This orange box on top here is actually an integrated GPS, an inertial sensor, and also it has a 480, I think, 500 gigabyte data collection hard drive or flash drive on here. So when you're talking about size, weight and power considerations for a drone such as we have here, this is an incredible advantage. We have a tiny device with its integrated data collection and GPS. Headwall Photonics worked with Leica Geosystems. You know Leica from the microscopes perhaps. Leica Geosystems developed with Headwall this drone here, this orange beauty, that has everything on it that you need. So it will have this nano hyperspec, GPS, inertial sensor, LiDAR, data collection unit, which is this tiny orange box on top. And a great advantage to this system is its ability to, you can enter the GPS coordinates, a polygon, a square that you'd like to collect data from, so in your field, and press go, and the drone will fly off and only start recording data once it's over the area you specified. So you gain great advantages in time saved, in collecting just the data that you want, and cutting down all the complexity about getting rid of data you don't need. It does auto rectification, which I'll show you in a second. It's a very good integrated package ready to go. So we'll just go on to show some examples of what these hyperspectral cameras are used for. Here we have a hyperspectral data cube collected from the ESA Flex program. At the top you can see the red, green, blue simulation of what it would look like to our eyes. We have lots and lots of information. We can reproduce an RGB image, and we can see it's a mixture of fields with crops and soil and roads and all sorts. And from this data, after processing, we have a decision made, or we can highlight and quantify areas in this image and show exactly where the crops are growing and where there's just bare soil. And depending on how well these crops are growing or how healthy they are, perhaps, we can color code. And you can see these false coloration images quite a lot when you're dealing with spectral imaging. Once you've collected the data, you process it and output a false color-coded, false contrast image with a lot of information there to make decisions on. So another example perhaps could be from the Ariana project. On the left, we can see what the scene would look like to us. Just a patchwork of green and brown. You can make out some roads and trees, fields, etc. But using this kind of the powerful post-processing on these hypercubes or data cubes, we can start to classify and quantify the areas in the image, whether they're grassland, woodland, roads, playing fields, buildings, all that sort of thing. So this, for example, could be very useful to the military for finding camouflage tanks, things that might look invisible to our eyes because it's a certain shade of green, but in the hypercube and the processing, you can actually highlight and find whatever's in there. Taking it down back to the ground, this slide is really to illustrate 2 points here. First of all, that in the last few years, sort of 5 years or so, the equipment and cost of the equipment and ease of use of the equipment has dropped dramatically, making it much more widely available to research groups that are interested in doing this down in the field or in the lab. So this image here is right up close and personal with some wheat ears, and they've used a hyperspectral camera and imager to take an image and then quantify the degree of infection of fusarium on these wheat ears. So you could then use this calibration, this data, to go and feed into a hyperspectral drone flying over the field that they could then quantify over your entire field how badly infected by fusarium it is. Another note to point is that you get a degree of infection. It's a quantify. It's not just is it infected or not. We can tell how infected it is. So these guys in their picture have indicated the degree of infection and color coded it again with this false contrast, false coloration that you always see. Another nice application down on the ground is in chlorophyll content. So here these researchers have used an imaging spectrograph to image some leaves up close and personal, and based on the spectral signatures of different pixels in the image, you can retrieve a lot of information about the chlorophyll content. So I'm going to start to move on to talking about multispectral imaging. I'll just quickly point out some differences, similarities. It is the same idea where you have an image with many spectral data points behind each pixel, or if you like, another way of looking is a data cube with many hundreds of images. So with hyperspectral, you might have anything from 200 to 1,000 images in this data cube. Multispectral imaging it's just the idea that you have far fewer of those images, far fewer of those spectral data points in your data cube. So the data files are really a lot smaller. So in hyperspectral imaging, you'd have gigabytes and gigabytes of data. Multispectral, it may only be a few hundred megabytes. The analysis is therefore quite a bit more easy with multispectral imaging, and there is dedicated software out there, particularly rather with the VideometerLab. I'll show that in a minute. The hyperspectral imaging, you do get these very high resolution spectral signatures. You get all this data, and that's absolutely fantastic. You can retrieve a lot of analysis from there. And multispectral imaging, you've got a much lower resolution spectral signatures. Now you might think it's therefore a poor man's hyperspectral, or it's not as good, But in a way, you'd be quite mistaken there, and it's kind of horses for courses. Just because it's multispectral imaging doesn't mean it's really any worse. It may actually be an advantage to you. It's much easier to analyze, much quicker to analyze. And really, in a lot of situations, you don't need 90% of the information in a hyperspectral data cube. You might only need what's in a multispectral data cube to tell the same thing. And I'll illustrate that now with a kind of an example. So if we had a leaf, and we know this leaf is a mixture of chlorophyll A and chlorophyll B, and we spent time in the lab using a spectrometer to find out the absorption or reflectance spectra of each molecule separately, chlorophyll A and chlorophyll B, and we can see them here, nice smooth spectra. With multispectral imaging, what we can do is just interrogate or sample those spectra at certain points. So here is just 4 points along the spectra, and we can actually see that chlorophyll A and chlorophyll B are quite well separated in just those areas. They have their own unique pattern, their unique spectral signature, even though it's a low resolution 4 data point spectral signature. They're quite distinct, and we can actually chemically identify and spatially locate those separate, very closely related molecules over the surface of a leaf. So although we've drastically cut down the amount of data we're collecting, we're still getting a lot of analysis value from it. And to take it back to, for example, the example I showed you earlier of fusarium infection on a wheat ear, we can actually do this same thing with the VideometerLab. So that's just a multispectral imaging system. It only has 19 data points in each spectrum. But here we can see a Petri dish. Well, that's been masked out, the Petri dish, but a Petri dish with the grains of barley and fusarium infected barley. And we can run an analysis to actually find, locate, and quantify the degree of infection on these barley samples very quickly and very easily. And we can train the algorithms and save them, save the light settings, and do this again and again in a very objective and repeatable manner. So here's an actual application over in the Danish Molting Group, I think it is, where we have, I'll play this video, but first I'll point out we have the integrating sphere of the VideometerLab here, a little conveyor belt that's feeding grain into the imaging area just underneath, and a hopper that you can fill up. So what happens is it just vibrates and distributes some grain onto the conveyor belt, which then goes under and is imaged. The blue is like a green screen, if you like. The blue background of the conveyor belt is like a green screen that can be masked out easily by the software. At the moment it's pausing between each image acquisition just so you can see clearly what's going on, but in reality the image acquisition time for a VideometerLab anywhere between 5 and sort of 10 seconds depending on how many of the LEDs you need to use. You might not need all 19, you might only use 5 or 6, and so the image acquisition time will increase, the data file size will go down, and once it's gone under, it goes past in the conveyor belt and is just collected at the end. There you can see the entire system. So just normally the VideometerLab on its own is all you'd need, and you would have to manually feed Petri dishes underneath and take the images. But with this you can get a much more high throughput system going. Here's a brief schematic of the VideometerLab 4. So what we've got here is an integrating sphere. All that is a hollow sphere about a foot across, a foot and a half. It's painted white on the inside, and it's got LEDs all around the inside circumference. So there's 19 LEDs all around the inside. This is a motorized raise and lower, so it goes up and down, and you put your sample here. At the bottom, the sphere descends and encloses it inside. The LEDs shine in turn for a very specific amount of time depending on your light setup, and the camera up top collects the reflected light from your sample. Very easy, quite intuitive, it's very easy to understand. Ignore for the moment this emission filter wheel. I'll explain what that's about. That doesn't come as standard, but it can be very, very useful. The LEDs provide what's called active illumination, so we can really very precisely control the lighting in here. We're not reliant on the sun, for example, and it doesn't get cloudy, or you don't have a different times of day, whether it's noon or sunset. We can very precisely control the lighting conditions between UV 375 nanometers all the way up to infrared 970 nanometers. There's a 6 megapixel camera up the top, and that acquires an image each time the LEDs shine in turn. A lot of people, just a quick note, a lot of people think, well, 6 megapixels, that's not that great, is it? Because my iPhone has a 15 megapixel camera. It does, but it's not really comparing light like for like. For example, 15 megapixel iPhone camera, you've actually got 5 megapixels each for red, green and blue. So they're separated. The actual sensor has a filter printed on top of it to collect red, green and blue light separately and create an image that looks pretty to us. But what we have with the VideometerLab, a 6 megapixel camera acquires an image at each wavelength. So in effect, if you're going to compare them, this is in effect like a 120 megapixel camera. So a 6 megapixel image at 19 different wavelengths, it makes like 115 megapixels. So once it's collected, there's 19 LEDs, it collects an image. For each LED, you have 19 images per data cube, or if you like, 19 data points per pixel. The filter wheel, I'll quickly mention, it's an optional extra, but that would allow you to look at multispectral fluorescence. So we cut out the reflected light and only allow through fluorescent light above the wavelength of the LED. And so that could add, in effect, up to 27 extra multispectral fluorescence images. 27 plus 19, you can have a 46 band or 46 image data cube from this setup. For those who are interested, if you can download this presentation later, these are the wavelengths of the LEDs in the VideometerLab. They start in the UVA, so that can be looking at fluorescence or GFP, excitation, and they go at intervals, don't have to be regular intervals, just intervals through the visible and into the infrared. It stops at 970 because of the limitations of silicon sensors. You'll find with the VideometerLab and a lot of hyperspectral cameras, you'll find that they will often go from 400 nanometers to say 1000 nanometers, because above 1000 nanometers, that's infrared, silicon is transparent, and so you have to move on to in-gas sensors. So it's a different technology to look at far infrared light, but we can get a lot of information just using the what's called the VNIR, visible and near infrared region, between say UV in 350, 375 up to about 1000. So why would you want to use the VideometerLab for multispectral imaging in the first place? Well, it allows you to do very high throughput, sort of semi-automated workflow imaging using multispectral imaging, which is much better than normal color imaging, which is always non-destructive, objective and very repeatable. And it's in fact very flexible and general purpose. So for general purpose quantification of features, I've put in quotes, in a petri dish sized image area. So the image it takes is of a petri dish. It's as if you're looking at a petri dish from about arm's length, and we can measure and quantify both spatial and spectral features. So spatial can be area, size, shape, length, and spectral features could be the color, or indeed the chemical content of our sample. So who's actually using this? Well, in the plant science arena, there is one up at the John Innes Centre up in Norwich. They've had one about a year and they're still developing applications for it. For a few years, they've had ones over in Wageningen University in the Netherlands, Chavez in France. I know Syngenta in Holland have one, and pretty soon Rothamsted Research will have theirs installed in a month or two time. It's just being built now. Some examples of not plant science customers just in the UK. We have Boots, who use it to look at makeup or makeup wear. BP Castrol, using it for metal corrosion experiments. The Home Office has one at their experimental research labs to look at fingerprint development and fingerprint analysis, and Procter & Gamble have one in Egham for looking at counterfeit packaging. So widely different applications, really very versatile, and even in plant science it's very hard to say what a certain research group might want to use it for. It has all sorts of applications. So I think I'll go on and show you and talk a bit about those applications. In the plant science arena, so these are groups abroad that are using the VideometerLab. It has very wide applications. You can have a look at Google Scholar. Circadian chlorophyll rhythms, cultivar discrimination, screening seeds, transgenic seeds. There's quite a good example just recently in plant genetics, the paper Plant Genetics, or the journal rather, on using these multispectral images for seed bank, gene bank accessions. So we can relate the genes and the phenotype in a very close manner and have a real valuable database of these multispectral images that you can collect easily and store them away for future use. Various applications examples in general, so for grain and seed variety, disease detection, there's lots of malting applications, germination and hydration. In food, we can look at quality control, authenticity, contaminant detection. As I mentioned, the FSA, the Food Standards Agency, is very interested in this at the moment. The Lab of the Government Chemist, LGC, is trialling a VideometerLab 4 to look at food authenticity in various scenarios. Pharmaceuticals is becoming a very popular application. You can look at things like coating and API distribution in tablets. Materials and surfaces, grading, I've used corrosion here as an example, but really any kind of surface analysis is applicable. A nice little trick is agar plate colony counting. So we can see a couple of images here, perhaps a bit small to see, but an agar plate with two different kinds of colonies here, and it can tell the difference between the two and count them separately and quantify them separately. Forensic analysis is another application, of course. The Home Office has one for fingerprints, but we could also look at bodily fluids or question documents, counterfeit documents, inks, that sort of thing. And because we are looking at the visible and near-infrared region, we can actually use the information to look at color as we would perceive it. So C-Lab color quantification and simulation of different lighting conditions, if that's important to you. So here we're going to just quickly move on to a few different actual images taken from the VideometerLab, just to illustrate the sort of data you get from it. So this is a purple Snapdragon. It's just resting in a roll of sellotape here just to hold it upright. This is the RGB, or red, green, blue image generated, so what it would look to our eyes. And these are the 19 images that it's taken in each individual wavelength. So starting in the top left is the UV, and then blue, and then goes through the green and visible, red, and into infrared region. And this has just been false colored with a jet coloration to enhance the contrast for our eyes. And you can make this into an animated GIF and see where this hot spot right in the middle is of highly reflective UV and blue light area, whereas as you go on into the infrared, you can see there at the end of the GIF, the whole flower reflects equal amounts of infrared light over the whole surface. There's no distinguish. A nice example, a lot of people use Arabidopsis as a model organism. Here we have a transgenic Arabidopsis plant on the left, GWD genotype. I don't know what GWD stands for. And on the right we have a wild type, and you can see to our eyes they both look green. We can't really tell any difference. But in the software, what I can do is highlight groups of pixels together. So on the left hand side, all these red pixels I've highlighted as a group of an example group, and on the right hand side all the blue pixels. We're going to group those together and look at the spectral signature, and we can see that for the GWD phenotype in red here, although it's very similar to the spectral signature of the wild type in blue, it's got a very similar shape. We can see that in the infrared region up here, So we've got the wavelength down here. This is the infrared region above sort of 700 nanometers. It reflects far more infrared light, and we can't see that. That's invisible to our eyes. So we can use that as an indicator, perhaps, when we're screening plants to see if we've been successful in transforming them. We can use that to see if they are transgenic or not. One of the most fruitful applications for the VideometerLab has always been grain and seed analysis. So here's an example we have of simply some wheat and barley grains in a dish. First, we use the software to teach it what grain in general looks like, so it can separate it from the background. So wheat and barley are the same thing here to the software, but we can also teach it to tell the difference between wheat and barley. It's just an example application. Of course, we can ourselves, but this shows what it can do. So we've got wheat and barley highlighted in red or blue. And once we've trained the software to do this, it can isolate the grain from the background, a bit like a green screen when you're watching the weather, then separate the grains out and line them up in what's called the Blob tool. So here what it's done is isolated each grain in the dish and lined them up all in a grid and generated lots and lots of information on it. So things like the area, the size, shape, length, the color coordinates, hue, intensity, all those. And we can add to these features as well. We could create an index of fusarium infection, for example, a score between 0 and 1 of how badly infected this grain is, and that number would have appeared down here. When we could report, create a PDF report or export that data to Excel for further analysis. We can line them up, color code them, classify them even. And this is actually what the John Innes Center has been using it for, or developing this application. They haven't published anything yet, so still in that development with Videometer, where they have the VideometerLab instrument. They feed it a dish of germinated wheat, and it will take an image and separate the grains out, line them up, and then it's classified each grain on whether it thinks it's germinated or not. So I think here is blue is germinated, green is ungerminated, and red is upside down, or blue is upside down, and you can't tell. But it's been classified here, and although this may not be 100% accurate as compared with a technician physically counting them in a Petri dish, it is a lot quicker, and the VideometerLab doesn't get bored. It never changes its own criteria for counting what's germinated or not, and as long as it's close enough to the true answer, say within 5% or even 10%, it means you can get through thousands of samples in a couple of weeks as opposed to a couple of months. Another nice application could be in pathology, so in leaves, or of course seeds, as I showed earlier, but here we have a leaf, and actually this was taken at the John Innes Center quite a while back as just a bit of a suck it and see image, where we know that the top half of this leaf has actually been infected with a pathogen. It's been deliberately inoculated. So what we've done is I've cut out a small section and again highlighted pixels with the software to give it two conditions, two classes. Above is infected and below is not infected. And we've trained what's called an NCDA algorithm. It's just one of the statistical, multivariate statistical transformations, these operations. You don't have to understand how it works. You can just use the drop-down boxes and create these sorts of models in about a minute. It's very, very simple. And what it can then do is go through the rest of the image and classify the rest of the pixels in an RGB image and turn it into a transformed image showing an index of infection here. So we can see that the infection is spread a lot over this top half. You can just about see a little damage of the inoculation. In fact, if I go back to here, you can see the points where it's been inoculated, highlighted. Very hard to see in an RGB image. Can't see it. But it's very obvious once you've done these models. Another application was root disease. So we've got a very clear agar with some seedlings growing in it, and they have disease roots here. And again, all I've done is trained an algorithm, this NCDA, to tell the difference between root disease and anything else. so it's just the root disease is important and interesting here. And again, use the Blob tool to pull out those areas and generate a whole load of data on it. So you could imagine doing a time series of this, taking images every half day or something and tracking how the disease or infection spreads through the roots. And you can export this to Excel and keep this data for further analysis later. Finally, a rather nice example of high throughput seed analysis is with oilseed rape and charlock, or wild mustard seed. What we have here is a petri dish full of oilseed rape and charlock seeds, wild mustard seeds, and the charlock seeds are a little bit smaller. You can see that, but you know, it's really hard to tell exactly which one is an oilseed rape and which one's charlock. So how much do we pay the farmer for this? So under the blue light LED, this is at 450 nanometers, blue light, stuff light we can see, there's really no difference. We can't see what's going on. Now if we use infrared light though, this is at 780 nanometers, so beyond what we can see, those charlock seeds, they really light up. They reflect a lot more infrared light than the oilseed rape does. This is the same image but false colored in this jet coloration just to enhance the contrast, and the oilseed rape really stands out like a light bulb. And we can see, for example, charlock seeds that are the same size as an oilseed rape, so you might mistake them for oilseed rape, but in 780 nanometer light we can tell the difference night and day. And then we can build a model based on this and run it on high throughput petri dish, or you saw the auto feeder earlier. We could use that to feed it oilseed rape and admixture and then classify and quantify the area of oilseed rape and admixture in that little area there. Now, rather than do this very manually each time, once you've developed these models and saved them with the light settings, so it's reproducible every time, you load the light settings, load the image analysis recipe, and you create what's called a session. And we have a session window here. This is just a double-click icon on the desktop. This window opens, you press start, and it will just start taking images, or it will just look to a folder full of images you already have and then analyze it using the algorithms that you've built in. So we can see here we've got a mixture of oilseed rape and charlock. It's distinguished oilseed rape from the charlock and quantified each one separately. And I've just got a very short video here of the session, one of the session models in action. I've sped this up by 2 times, so this is twice the speed it normally goes at, and it's actually just looking at an image full of a folder full of images I already have, rather than taking images live from a VideometerLab, but that is of course what you can do. I'll just press play here. So we go up to open the session manager, press F12 to start. It's looking into an image full of a folder full of images, and you can see what it's doing is it's analyzing the image, spitting out a color photograph equivalent, and then an analyzed and transformed image at the bottom to show the areas that it is counting as oilseed rape versus admixture. And it's generated a table of results that is also exported to an Excel sheet that's auto-saved in the same folder as all the images that have come out here. So it can be very, very fast and very, very easy way of grading admixture and oilseed rape or similar situations. So sort of coming towards the end of our presentation here, thanks very much for listening. To reiterate, the VideometerLab 4 and spectral imaging in general is a very powerful technique. So we've got high throughput plant and seed phenotyping that's available to technicians and non-experts now. That's where we've got to. So rather than the expensive NASA kind of systems, we can all do it ourselves now and get a very nuanced human-like analysis. There's very sophisticated tools for both the power user and a novice. So the technicians, for example, could use those session recipes. They don't have to build models. They can just press go and it will generate analysis results. It's very fast. It's non-destructive, which is great. You can then do all the other tests to your heart's content. If you want to do destructive testing, you don't really need any sample preparation at all either. There's various links in this presentation, and you can download it later. Click these links. I'll just quickly point out these videos you can watch. If you click on these links in the presentation, it will take you to our YouTube channel here. Here you can see all the videos we've uploaded if you click on uploads, but the one in the top left at the moment, the most recently uploaded, is a really short, about 1 minute, 1.25 minute video on plant and crop phenotyping with the VideometerLab specifically. I just hear myself in my earphones, and this is just a nice simple software demonstration rather than showing you right now. I think you can have a look at this and see the sort of results and how easy the software is to use from there. And please forward that on to any colleagues that you think might be interested, or if you have application ideas, please do get in touch. If you want to see a bit more in-depth analysis with the software, have a look at this Oil Seed Rape and Charlock analysis video. It's actually a playlist. If you go to Creative Playlists, you can see one of them here at the top right. It's a 4-video series, each about 5 or 10 minutes long, that just goes from the very basics. We have Oil Seed Rape and Charlock in a dish, takes you through the basics of getting data and generating models. I hope you find it interesting. Okay, so I think rather than go on to hardware, software options and post-sale support, those are there if you want to have a look at them later. I think what I'll do is open the floor to any questions that people might have. If you'd like to have a look over on the right hand side of your screen, you should see a question panel where you can type some questions, so please go ahead. Okay, so I'll just open the questions. Okay, so we have a question here from Oliver. What about 96 well plates? We have done some imaging with the VideometerLab 4 with plates. Sometimes you need to mess around with the focal plane. You usually get some quite good results. It can be used with 96 well plates. The only drawback sometimes is that the wells can be deep, and so the imaging focal plane might be a bit out of focus. It's perfectly possible. It might well be more suited to perhaps hyperspectral imaging, where you can set the focus at the bottom of the plate and scan in a push broom manner. You can have these kind of lab versions of a starter kit where you have illumination and a translation stage, basically a conveyor belt, moves the sample underneath, and you can collect a hyperspectral data cube. I will mention a slightly separate technique that we actually have is Raman spectroscopy for 96 well plates. So Serge Raman, we have an instrument from DigiLab where you can put a 96 well plate and it will give you Raman spectra for each well. Perhaps it's worth us having a bit of a chat later. Oliver, I'm going to talk a bit more about what you'd want to image in these plates. Yeah, we have another question here from Jiju. If you would like to try out or have a demo of any of this equipment from Headwall or VideometerLab, please just get in touch with us. The European sales manager for Headwall Photonics is Scott. Luckily, he lives up in Glasgow. He's the one who would have demonstration equipment, so we would be able to arrange a demonstration at some point depending on what your application is, whether it's airborne or in the lab. So please just get in touch with us and we can talk about that separately. Email addresses and all the contact details will be in this presentation, which you'll be able to download. We'll be sending out a link after the presentation once we've ended with a link to this presentation, a link to, we've recorded this presentation as well. You can watch it again or send it to your colleagues at any time. Oh, we got one more question here. How long does it take to acquire the data cubes? It takes about roughly 5 to 10 seconds with the VideometerLab. With the head wall gear, if you had a starter kit down in your lab, so you're collecting data cubes of something in your lab, it will take about the same time as well. The setup is a little bit more complex, but you should be able to set up once and collect data cubes within 30 seconds or so. Just check for questions here. Okay. Yeah, we have another question here. How open is the device? Can you connect it with a computer? So the head wall, the VideometerLab will run with any PC. It has its own dedicated software for acquisition and analysis. I showed you some of that. You can see some of that in the video. It just needs a very powerful computer. It just needs a lot of RAM to work. Similarly, the head wall equipment, you will need a very powerful computer, often with some quite specific hardware, network cards, frame grabbers, that sort of thing. Head wall and Analytik, we can supply that computer as well and guarantee it will work with the head wall device. But you are welcome to use your own computers as long as they've got enough RAM, enough processing power, and crucially those certain bits of hardware like the frame grabbers that are needed to work with the head wall cameras. And the head wall cameras actually come with a few different kinds of camera links, like base link or camera link, depending on your hardware setup. Okay, does anybody else have any final questions? No? Okay, that's fine. Thanks everybody for attending our webinar today on spectral imaging for plant and crop sciences. I hope you found it interesting and informative. Please do get in touch with us anytime if you'd like more information about the technique or what it could be used for. If you have a particular application in mind, it's probably possible. It's a pretty amazing technique. I very much enjoy discussing this sort of thing with researchers. So I hope to hear from you all soon. Thanks very much. Take care and goodbye.